So for those of you who don't know me, I'm Jim Perkins, and I'm director of the graduate program in medical illustration here at RIT. And we have 5 presentations tonight. Our second year graduate students who are finishing up their thesis work are going to present their artwork and in some cases animations. So I'm going to ask folks to mute themselves if you're not actually speaking, because I hear some background noise. So out of respect for the presenters, let's all mute if you're not actually talking. And without further ado, let's go ahead and kick things off with our first presenter, Danielle George. Go ahead and share your screen.
Well, hey everybody, can everybody hear me? I'm assuming.
I can hear you loud and clear.
Okay. Move the little toolbar out of my way. So hi, like Jim said, I'm Danielle, and my thesis is an illustrated guide for veterinary students that focuses on the more difficult aspects, things to learn about the spay procedures in dogs and cats. Ooh, seems being a little laggy. Okay, there we go. So today, I'll be briefly going over the surgical procedures and related anatomy and explain my purpose and goals for my illustrations and go through my process. And of course, what everyone wants to see, my final illustrations. So the ovariohysterectomy is the same thing as a spay. It's so in dogs and cats, it's the full removal of the ovaries, uterine horn, and uterus. It is an elective procedure that has many benefits to the individual pet, as well as helps regulate population size. So I know we have a lot of people in the audience that aren't as familiar with dog and cat reproductive anatomy, so I decided I'd do a side-by-side comparison of both human female anatomy and dog female anatomy. And one thing that you might notice that's different about the dog is that it has a split in the uterus with these 2 uterine horns coming out, and it comes way further into the abdomen. And that's basically just to accommodate for the larger litter sizes that they have. So I'm going to go through the surgical procedure really briefly, just to kind of have everybody on the same page. And so first things first is the skin incision along the abdomen. Then we have finding the uterine horns and pulling them up. It's normally done on the right side first. Next is breaking the suspensory ligament, and that allows better access for the ovary. Next, there's making a window in the broad ligament, so then the clamps can be attached to the ovarian pedicle. Following that is the ligation of the ovarian pedicle, and then detaching it. Remember, there's 2 uterine horns, so you got to do the same thing on the other side. And then we have, after both the uterine horns are up, they get retracted caudally, and then the ligation of the uterine body and vessels happen, and then the uterine body is detached. And then finally, just suturing up your abdomen. So the big question, why create more illustrations of the spay procedure? Because we have a lot of illustrations that exist now, but I feel like the spay procedure is generally the first open surgery that a student will perform, and it serves as a core foundation for successful completion of other surgical procedures. And almost all veterinary schools in the United States require that students do perform a spay procedure, and it can be really daunting. So I did want to create illustrations that would be very easy to understand for them. And so to get a better understanding of these needs, I talked with my content advisor, and I see Ari's here, so I'm really happy. to kind of get an idea of what students need in illustrations, I also interviewed surgeons and veterinary students when I visited the Learner Clinic at the Cummings School of Veterinary Medicine. As well as I also did some interviews with some past classmates of mine from UC Davis that are now practicing veterinarians. So let's kind of go through some of the goals of what I wanted to achieve with my illustrations. The first one being compare the female reproductive anatomy between both dogs and cats. And I especially wanted to emphasize the depth of the uterine horns, as well as how lateral they fall within the abdomen. The next goal was to see where the different skin incisions go, depending on the age, species, or clinical history of the patient. So here I have an adult dog, a puppy, and a cat, or like a kitten. They have pretty much the same incision site. So then another goal being finding the uterine horns. And this can be done with either a finger or a spay hook. So trying to incorporate the anatomical images and include the relevant information. And so next we have the breaking the suspensory ligament. And this has been traditionally kind of one of the hardest things for students to visualize because it's really hard to see when it's going on. The next goal is to show the 2 and 3 clamp techniques in surgical context. Next is the ligations that can occur, and these are different depending on the needs of the surgery. So a little bit of my process of how I started this project. So I definitely started with doing a deep dive into the research before I did any drawing, just to make sure that I understood all my anatomy. I looked in a bunch of different anatomy textbooks and surgical atlases. I was able to, like I said earlier, visit the Learner Clinic at the Cummings Veterinary School of Veterinary Medicine. And I got to do it twice during the course of my project and observe spay procedures live for both dogs and cats. And during my observations, I was able to sketch, take pictures and videos of the surgeons performing the procedures. And I was able to speak with the surgeons and veterinary students about what they would have liked to have seen when they were learning this procedure. Additionally, my content advisor did provide me with some surgical teaching videos and DICOM data of 2 intact females that happen to be getting MRIs done at the vet school. And speaking of which, here's some examples of that DICOM data visualization. This is in 3D Slicer. You can see that, I can manipulate the 3D body around and remove tissue. And then here's an example in Horos of getting that nice sagittal cut. So now just like the sketching, just being a very important part. I like to start with pencil and paper. Call me old school, but works. I mean, I like to test that I understand the anatomy before kind of figuring out what sort of illustrations I'm going to create. And I have some examples of me going over some of my more refined sketches with some tracing paper. But then when I do happen to sketch on an iPad or such, I typically will use Adobe Fresco or Procreate. And I'll always take these illustrations that I've created using that sketching method and put them into Photoshop so that I can further refine them. And so then I also have a bunch of surgical photographs that I took through the course of this process. So I did go in through Lightroom and adjust the lighting just to make sure that they were nice and clear. And with that, I did want to give a little warning that I will be showing some surgical images. So if you don't like blood or surgery, turn away for a couple seconds. It'll go real quick. So here we have an example of the ovarian pedicle. It's this beautiful little red squiggly line. And this is in a kitten. You can see how it's important that you need to ligate these vessels because they are rather large in comparison with the size of the structure. Next, couple things I want to point out. Here's breaking the broad ligament over here and some of the clamping techniques. Those images before were taken with a nice DSLR camera. Surprisingly, these images that you see here, these were actually taken with my cell phone camera. I did, to achieve actually getting an image, have to use the manual settings in my camera, which that was kind of a fun experiment. But yeah, there's one image that I really want to point out, and this is this one right here. This is of the surgeon breaking the suspensory ligament, and I wanted to point it out because it's really hard to see what she's doing. So it kind of explains why students might struggle with visualizing what's going on during that. And then without further ado, here is some of my final illustrations. So here is some of my anatomy images. Once again, trying to create images that were really emphasizing how deep the uterine horns fall within the body wall or the abdomen, and how lateral they are to the body wall. Here is the skin incisions again, and just kind of comparing, depending on the species age or whatever, where the incision placement goes. And I wanted to make sure in these illustrations that I included the umbilicus and the pubic symphysis. I also have the linea alba drawn in and the teats for some landmarks. So then in locating the uterine horns, I thought that it was important to show the hand in context with the body. So this shows the depth, or going along the body wall with the finger. This next image is showing the depth of how deep the finger is going and grabbing. And just to give you a little, this is more the proportions of in a puppy as opposed to a full-grown dog. And then this image is of a cat with a spay hook, and you can see how far in and lateral the hook goes. Next we have the suspensory ligament, or breaking the suspensory ligament. And most important part being showing the finger running along the suspensory ligament and feeling all the way up, and then grabbing the ovary and pulling it up and out to create tension to allow you to break the suspensory ligament. And then now I have a bunch of series of surgical steps of first clamping techniques. So this is with the ovary out of the body and the window already made, adding the clamps. We have the 1 clamp, the 2 clamp, and this clamp is moved so then the ovary does not fall back into the body. And then with your 2 clamps in place, bringing in the needle driver to bring in the suture to begin the ligation. Okay, so here's some examples of ligation techniques. First, we have the circumferential ligation technique, which is, you see that pretty commonly with spay procedures because a lot of times this is done in puppies and small kittens, and they don't have very big vessels, so they don't require much more than a circumferential. So here we have the needle driver coming down and underneath the 2 clamps and through the window to grab the suture. The suture is then brought through the window and underneath the clamps. And then a surgeon's tie is begins. And then just wanting to emphasize using the clamp to, when you tighten down your suture, to make sure that your suture doesn't slip around. And then, yeah, so another one of the ligation techniques is the modified Miller, and this is oftentimes done on the uterine body, so I chose to depict it like that here. Once again, putting your 2 clamps on, and needle drivers coming down and under, the suture coming in. And then the suture gets brought under the uterine body and under the clamps, and then is looped around the finger. It is then brought down and around and looped over the finger again for a second time. And after you have 2 loops, then it's brought over the 2 loops, and the needle driver is put through those 2 loops, and the suture is then brought under and through. And then you should have 2 parallel lines of suture running. And then once again, just like in the other ligation, using that handy clamp there to make sure that your suture isn't sliding around and tightening. So transfixing suture is the only one that I've shown that uses actually the needle that then is pierced through the tissue. And then a knot is tied, and then following that knot being tied, the suture is wrapped around the uterine body again, and then you have your usual surgeon's throws that happen. And with that, we have the end of our spade procedure. And here's our lovely ladies who just got spayed and are in recovery. So thank you so much. That's my presentation. I really wanted to take just a second to thank Jim, Craig, and Glenn for being awesome. And thank Ari again, because you are awesome and she's here. It makes me feel really happy. I also wanted to thank Kip for being here, and thank you for looking at my illustrations and giving your comments. That was really helpful. And I don't know if Allie Buck is in attendance today, but I really wanted to thank her too, because she put me in contact with Ari and just made this thesis all possible. So thank you so much. That's what I got.
All right, thank you very much, Danielle. That was great. I think we're, in the interest of time, we're going to jump right ahead to our next presenter. If folks do have questions, maybe we can hold them to the end just to make sure we get through all of the presenters in a timely fashion, and then you can ask any questions at the very end. Or if somebody happens to go extra short, then maybe we can make up a little time, but it's mostly because I blabbed on for a couple minutes at the beginning. So Catherine, next presenter is Catherine Lysing, if you'd like to share your screen and take over.
All right. Okay. Can everyone hear and see the screen?
I can hear you and I can see it, so hopefully everybody can.
Cool. Okay, so hi, my name is Catherine Lysing, and today I'm going to be presenting my master's thesis, which is about carbon nanotubes, an introduction to the future of immunotherapy. Just to quickly go over what I will be talking about in this presentation, I'm first going to start with a little introduction about myself for those who don't know me, as well as my background and my content advisors. I'm going to be discussing some of the scientific background, explain what carbon nanotubes are and how immunotherapy applies to them. I will be discussing my target audience, how I came up with the process and conceptualization of my animated film, and the process of gathering research and information for that. I will be discussing the steps of what needs to be done post-graduation and presenting my references and ending with some acknowledgements. So just a little bit about me. I'll quickly go through this because I know we're a little fast on time. I graduated with a BFA in animation from Damon University in Buffalo, New York. Since then, I've had an incredible experience here at RIT. I have worked as a GA, I've worked as a tutor, and I've also done a couple of journals for scientific papers. I am a member of the Association of Medical Illustrators and also a member of the Guild of Natural Science Illustrators. And after this presentation and everyone's presentation, if you'd like to see more of my work, there is more at catherinelicing.squarespace.com. So now to introduce my team, and I have a couple of people in the audience here as well. Going from left to right, I've been working with Dr. Michael Charlou, who is a professor at RIT and the Department of Mechanical Engineering. This is Dr. Ian Dickerson, who is a professor at the U of R, and Dr. Omar Bakht is the CEO of AGTC, which is also located in downtown Rochester. And he is also at the. So let's talk a bit about carbon nanotubes, what exactly they are. They are tiny circular-like figures that are composed of carbon within a aluminum anodized oxide membrane. And we reach the carbon nanotubes through a series of chemical vapor deposition, removing of carbon by plasma oxygen to expose the aluminum membrane, and etching through a selective removal through exposing the carbon nanotubes. And just to get a sense of how small these nanotubes really are, the membrane itself is about the size of a dime, which is 13 millimeters in diameter. These nanotubes come in thousands on the membranes, and they're about 200 nanometers in diameter, which compared to in millimeters is 2/10,000ths of a millimeter. So very, very small, but that is helpful for the ability to penetrate cellular membranes, which allows for genetic material to alter the nucleus of the cell and alter the DNA of the cell. And this is really important for genetic studies for drugs and pharmaceuticals. And specifically, I'm going to be focusing on immunotherapy, which literally is defined as therapy that relates to the cells in the immune system. And that includes lymphocytes, dendrites, neutrophils, etc. There's many different types of cells involved in the immune system. Specifically, we're going to be targeting what is known as CD4 T cells, which are also called helper cells because they are very helpful in immunity, because they have the ability to remember previous encounters with allergens. And because of that memory, it increases efficiency in fighting infections or targeting allergens. And this process can be enhanced with fighting more difficult implications such as cancers and more efficiently fighting illness and infection. So the plan. My goal is to create an informative and animated video that educates leukemia patients about this new modern treatment. There isn't much, they're very few and far between animations and illustrations that are widely spread that are about immunotherapy, specifically how AGTC is using carbon nanotubes. So that is why I decided to construct an animated video for patients to observe. My audience is adult patients, parents of patients diagnosed with leukemia, or any individual interested in learning about modern cancer treatment methods. So again, primarily the goal is to complete an animated film and have it viewed by people in my demographic. Post-graduation, I will be discussing with them what they think is most effective about the video and what's not effective, and be reviewing this information with content advisors to make the best video possible. So here's some of the photos I took throughout the year of working with AGTC. This is at one of the labs, and that's actually on RIT campus. And I had the opportunity to test out a membrane vacuum.
Recording in progress.
It's this very tiny vacuum that because the membranes are so fragile, allows them to move them very carefully, they will then be put into a furnace, which I took a photograph of here, which is also on RIT campus. And that furnace allows for the exposure of carbon, as you can see on the bottom. Their membranes are white going into the furnace and black coming out of the furnace. So coming up with the development of this animation, I really wanted to make this nostalgic with a modern twist. And what I mean by that is my target demographic being more involved in the 60s and 70s, I wanted to appeal to that animation style. So I studied works from Schoolhouse Rock, UPA Animation Studios to get a more stylistic approach, but also have a more modern view since we're focusing on modern technologies, which you can see on the bottom. That's an image that I use as inspiration from NTL Studio, which is a bigger studio that makes graphics for web design and journals. I also use some AI generated concept art, which was really a cool experience because this technology is so new. I use this simply to make my backgrounds and get some ideas of what type of props and things I could use to make the most effective and efficient animation style. So from there, I did sketches on my iPad using Procreate and some storyboarding apps. And I was able to take those sketches and put them into a 3D modeling software that's known as Autodesk Maya to create a realistic perspective of what these would look like in relation to the characters. And from there, I used Adobe software to make my flat stylized backgrounds. I also did a combination of Procreate and Illustrator to create concept art for my characters. For example, this is my main character, Dr. Kelly, who has a more neutral color palette, as well as my patient character. I tried to go for complementing palettes so that the two characters can balance well with each other. This is an example of how I will be making the animation post-graduation. This is an example of the character rigs. This is what is called a puppet rig, where you take joints from a software and apply them to a drawing, and you're able to animate it. I also did some simulations in Autodesk Maya. This is a sample from a scene that we'll see in just a minute from my video. I used what's known as a particle simulation to allow a randomized number of red blood cells, white blood cells, and platelets to go through what looks like a blood vessel. These big red circles that you see are lights, and the little green box that you see is the camera. So we'll get to see it in that perspective in just a minute. Now, what I'll be doing post-production will be completing my rigs for all three characters, fixing any animation errors, audio errors, and errors in the renders, and again, presenting it to the patients, getting their feedback, and seeing if my video is successful or not. And finally, the one important thing I want to focus on for post-production as well is accessibility and inclusivity. So I have embedded subtitles for people who are deaf and hard of hearing. My font choice is specifically chosen for helping people who have dyslexia or difficulty reading on a screen. And my color palette is adjusted for colorblind individuals. And just before I show my film, I just want to say thank you to the team at AGTC. I know that Michael's here. I want to thank Professor Jim, Craig, and Glenn. I want to thank all of my classmates who've helped me along the way and my friends and family. So thank you all very much for being here. And here's my video. And please keep in mind that this is a storyboard. It's not finished. And there is one audio change that you'll notice. And please let me know if you don't hear it.
A chronic leukemia diagnosis is most common for patients ages 65 and older. The patient may be experiencing common symptoms such as fatigue, weakness, and pain in their bones. A hematologist can observe indications of leukemia by looking at a blood sample from the patient. Within the bone marrow are developing red blood cells, white blood cells, and platelets. In a patient with leukemia, the affected white blood cells mutate and proliferate, taking up space and preventing healthy cells from maturing and multiplying. Because the cells cannot naturally keep up with and fight the cancer cells, the patient may be offered different methods of treatment. Most common forms of treatment include chemotherapy or radiation. While both are more affordable, they can easily damage healthy cells in addition to cancerous cells. This is where the option of immunotherapy steps in. Immunotherapy refers to therapy of the cells in the immune system, the cells that are responsible for fighting infection.
The carbon nanotubes must be exposed from within a membrane. The membrane is roughly the size of a dime, about 30 millimeters in diameter.
Through a process of annealing, applying gas and heat, and etching, they darken in color. The dark color comes from the expression of carbon, hence the name carbon nanotube. Thousands of nanotubes are then arranged in a grid-like base to place cells that are a part of the immune system. For example, this is a CD4 T cell, also known as the helper T cell, because they help to fight infections. Through the process of diffusion, genetic material coated on the nanotubes enters the cell. The material can alter the cell's DNA, giving information needed to recognize and destroy cancer cells. Today, professionals in the field are still working to understand how this process works and how it can be applied to other types of cancers. In a not-so-distant future, we may even be able to develop vaccines to treat cancers. While we may not have all of the answers now, the future continues to get brighter as we learn more.
Gonna unmute myself. Audio came through great, Catherine. That was great. Thank you very much. We're gonna move right along. Our next presenter, Mary Wen.
All right, can everybody see my screen all right? Can you hear me?
Yes, on both counts.
Awesome. All right, so my name is Mary Wen, and my thesis is visualizing visual neural pathways in virtual reality. Oops. So an overview of my talk today is I will be describing the role of this project for biomedical communication and why it's important. And then I'll talk about my research, and then I'll get into the materials and methods of it. And then I will conclude with the goals of the project and what I see for it in the future. So why VR? So as technology is expanding, with the rise of AI and ChatGPT, so is VR learning. I know right now, training in VR is especially growing. So because of that, I chose this topic, and I chose neuroanatomy because I've always found the brain to be super fascinating. It's so complex, and it's the only organ that can think about itself. And neuroanatomy has always been a challenging subject, especially the neural pathways. So my tool that I'm developing is for students to learn about the visual neural pathway in 3D space. And they'll be able to do this through different cross sections in the coronal, transverse, and sagittal planes. And they'll be able to see the 3D structures ghosted inside. My audience is targeted towards students taking courses related to neuroscience, psychology, medicine, or healthcare. But the tool is not limited to just students. It's open to anybody that's interested in neuroanatomy. So for my research, there are two key questions I focused on to guide it. The first question is, how effective is VR learning? And my second question are, what are the current modes of teaching neuroanatomy and the visual neural pathways? So from my research, I found that learning in VR is very effective because it allows a user to be completely immersed in the experience. And users are able to directly interact with the model, so they can rotate it in the X, Y, and Z axis, and they are able to see the different structures in 3D space, and it is accessible. On the other hand, the cadaver lab isn't ideal because not everybody has access to the cadaver lab. And in addition to that, cadavers are preserved using chemicals like formaldehyde, which may affect the texture of the brain and the color. So students aren't able to see the true color of the brain. And for web-based resources and textbooks, most of the images are flat, so they're not able to see the structures in 3D space like in VR. Right now, the current neuroanatomy tools are limited. So on the right is a photo I found. It's very abstract and schematic. I myself had issues interpreting it. So as you can see, it's labeled here as brain, but it's like, where is the brain? And there's a lot of information missing here. Are we seeing the view from the top of the body or from the bottom, right? So from my literature search, I found that clinicians now are worried about future clinicians because they think that there's a knowledge deficit in neuroanatomy. And because of that, there might be an increase in medical legal litigation and malpractice, which is very bad for both the patient and clinician. And for my scientific background, I used a combination of textbooks shown here. So I used the Human Central Nervous System, the Atlas of the Human Brain, and Clinical Neuroanatomy for medical students. And I watched different videos online and used web-based resources and also research papers. So here's a combination of everything I used in my notes. I also conducted a media audit. So the purpose of doing this was to see what's on the market right now and to take that information and make it better and see what works and what doesn't work. So on the left is a tool that's very similar to what I'm developing now. It's developed by the University of Wisconsin. And what I liked about this tool was that it's very straightforward. As you can see, it's not too distracting, but the brain was a bit inaccurate and it didn't represent the true color of the brain. And something I would really like to see is for the structures to be a bit more refined. So when sculpting it, making the structures a bit more realistic. But all in all, it was a good tool, but I think it could be developed more. And on the right was a tool developed by a university in Turkey. And as you can see here, the brain doesn't look realistic at all. There were a lot of anatomical inaccuracies, especially the cerebellum. So you can't see the cerebellum here, but when you watch the video, it's the base of the brain and it looked completely smooth, which is not right. And this was really concerning because the study was conducted, or the training was conducted for medical students. So what I did to start was I used Figma to wireframe the entire experience. The experience is very different from what it looks here now. What I originally planned was for the user to enter a neuroanatomy lab and have a message say, "Welcome to neuroanatomy," and pick up the brain. But what I've learned that it's best to just put the user directly into the experience. So I omitted this slide, and when the user enters experience, there's the brain and then there's a menu, so they're able to manipulate it easier. And for my materials and methods, when I was modeling the brain, I took into consideration 3 key factors. I want it to be accurate, and I want it to be detailed, and I want it to look realistic. So for the references, I combined textbooks, and I used 3D Slicer and the Brain Biodiversity Bank from Michigan State University. And then I used FigJam to compile all the images from the internet and the images from the textbook. And going over my first resource, which is the Michigan State Brain Biodiversity Bank, this was a great reference. However, I was only able to use it to aid me in labeling. And this is because So for their bank, they had 3 different specimens, which are 3 different humans. And as humans, our brains are all different, right? We all have different gyri and sulci. So because of this, I was unable to use all the MRI images that they uploaded to create a brain, because it would have come out not looking correct. So instead, to model the brain, I used the MR head data from the program called 3D Slicer. So some limitations were that the brain ended up looking a bit messy. And so I had to go back and refine it, and I'll explain them in the next slides. So I took the brain from 3D Slicer, the MR head brain, and I put it into a program called MeshLab. And what MeshLab does is that it decreases the number of faces in the brain, so it decreases the number of polygons. And this is really important because since the tool is going to be developed in a game engine, we want the experience to run fast. So by making a model too complex, by having too many polygons, it's going to slow down the experience. So this was a very critical step. And after I did that, I imported the brain into ZBrush as a low poly model. And here are some progression photos. Here you can see me sculpting it with a Wacom tablet. And then after that, I import it into Adobe Substance Painter, where I textured the brain and made it look a little bit wet and glistening, and then I added color to it. And here are my cross sections. I'm still in the process of illustrating everything. I have about 50 more to go, and I plan on going back and labeling it and using the Brain Biodiversity Bank to help me do that. And I'm using that on Procreate. So the pipeline summary for modeling the brain was I got the data from 3D Slicer, the MR head data. I put it to MeshLab, and then I put it in ZBrush to sculpt, and then I textured and painted it in Substance, and then I imported it into Unreal Engine. And for Unreal Engine, what I did was I used the program Autodesk Maya to 3D model all the assets, so the lab bench, and then the menu UI, and then the lab tray. So here are images of what it looked like in Maya, and then right here is what it looks like in Unreal. And here's a better picture of it developed. So this is what the tool looks like right now. I still need to add the UI elements. And Unreal Engine was it was a good experience. I learned how to use the visual blueprinting system. So here's a screenshot of what the material blueprint system looked like. And getting into the user interface, here is a sketch of the icons, and then to the right is the final. So here's the primary icons, and when the user hovers over it with the VR stick, what will show up is the text. And then once the user selects a button, then there will be a glow to indicate that the experience is going on. And my main goals and objectives of this tool is to enhance the user's understanding on the visual neural pathway and provide a comprehensive visualization that is accurate and also effective. And lastly, to increase the students' knowledge and ability to retain information about visual neural pathways. And what's super exciting is that my tool will be incorporated into RIT's new neuroscience curriculum. So this will be starting during the fall of 2023. And we will be conducting a study over the course of 2 semesters to compare test scores within students that are using this neuroanatomy tool in VR versus students that are not using it. So moving forward, I will be presenting this experience at Imagine RIT, which is in about 2 weeks on April 29th. And I plan on finishing the app by the end of the semester. And by fall, we'll conduct the research study and receive feedback from all the students and constantly improve the tool. So right now we're focusing on the visual neural pathways, but I'd really like to incorporate different learning modules and expand it, maybe expand it to how audio is processed. And here are my acknowledgements. Thank you to everyone that's helped me along the way on this list. And thank you for your time.
I keep forgetting to unmute myself. So thanks, Mary. That was very cool stuff. And moving right along, our next presenter, Joanne Park.
Hi, everyone. Can everyone see my screen? Okay, perfect. So my name is Joanne Park, and my thesis is on enhancing user learning experience of a new therapeutic approach for scleroderma using VR. So a little bit about me. I did my undergrad at Boston University, where I majored in biology and I minored in graphic design. My work consists of usually 3D modeling and animations, as well as realistic 2D illustrations. And if you would like to see more of my work, please feel free to scan the QR code or visit my website at parkvisuals.com. Before I begin, I would like to give a big thanks to my thesis advisors, Dr. Corinne Warzak-Kozak, Debbie Sanchez, and Craig Foster, who helped me guide me throughout my research. And a quick overview of my thesis. So recently, researchers at RIT have discovered a new therapeutic approach for scleroderma, also known as skin fibrosis, that involved the inhibition of TRPC6 channel. So to help spread awareness of their research and to enhance the user learning experience, an interactive virtual reality experience was created, as well as supplemental 3D animations. And so by engaging and interacting with the models, users can retain the information better and can improve their learning outcomes. These are the goals of my thesis. So first is to create 3D animations that allow the user to follow along the mechanisms of the TRPC6 channel and see how it's involved in development and progression of skin fibrosis. Next is to create a VR experience that allows users to interact with the 3D models while following along with the 3D animations, as well as engaging with the environment. And lastly, sharing the experience on my website. And these are the topics I'll be going over today. First, I'll be going over the channel and its relation to scleroderma, the effect of virtual reality in science education, the methods I took to create this, and finally, the results. So TRPC6 stands for Transient Receptor Potential Cation 6 channel. It's an integral membrane protein that alters the intracellular calcium concentration, and it's also responsible for a variety of tissue expression, physiological functions, and channel properties. The channel allows influx of both CH calcium and sodium ions into the cells via a pore between the 5th and 6th transmembrane domains. But TRPC6 is 6 times more permeable to calcium than sodium ions, resulting in an elevated intracellular calcium ion concentration. Next, I'll be going over scleroderma and TRPC6 effect of the tissue disease. So scleroderma is a complex systematic autoimmune disease that targets the vasculature, connective tissue producing cells, like fibroblasts and myofibroblasts. Disease manifestations vary from limited skin involvement to widespread skin and internal organ involvement. And women are affected about 4 times more than men. Unfortunately, it has a high mortality rate with a 10-year survival of less than 70%. The incidence of scleroderma is about 20 cases per million per year, and the prevalence is more than 250 patients per million in the USA. The exact etiology is not yet fully understood, although there's evidence that indicates that it can be activated through genetic factors and environmental triggers. Now, fibroblast transdifferentiation into myofibroblasts is necessary for cutaneous wound healing and tissue remodeling. But however, in scleroderma, abnormally activated dermal fibroblasts produce an excessive accumulation of extracellular matrix, mainly type 1 collagen, in the dermis, which results in progressive tightening of the skin. And research has shown that TRPC6 is involved in the development of skin fibrosis through the modulation of mechanical signals. So TRPC6 allows the influx of calcium into the cell, and this causes the activation of calcium-responsive protein phosphate calcineurin, which itself induces myofibroblast transdifferentiation. And the activated myofibroblasts then produce excessive amounts of collagen type 1 in the extracellular matrix, resulting in skin fibrosis. The pharmacological TRPC6 inhibition reduces the development of experimental skin fibrosis. So by using inhibitors for TRPC6, it can prevent the excessive production of collagen in the extracellular matrix. And here are some activators and inhibitors that open TRPC6 channel, as well as that close it. Next, I'll be presenting about VR in science education. So I did a lot of research of VR and in how it's being integrated into science education rapidly, and it's changing the way users learn. So research has shown that VR-based classrooms significantly improve students' academic achievement and science motivation. And has also been shown that VR training can decrease cognitive load on the learners and improve learning outcomes compared to traditional training methods. In addition, interactive VR environments can also help learners visualize three-dimensional shapes because the user is able to grab and rotate and examine the 3D models. And by engaging the learner's body, it can help them understand spatial awareness. And next, I'll be going over my methodology on how I created the project. So first, I designed the interface and the environment, and then the 3D assets and animations. And then I wrote the audio script, and then finally I created the environment in Unreal. So I wanted a very futuristic design, and I wanted a typography that was clean and bright, and I also wanted my color palette to have a lot of cool colors in it. Here was an early mock design of how I wanted my interface to look like, as well as my starting menu. And for the creation of my assets, I used Maya, Cinema 4D, ZBrush, and Adobe Substance Painter. So to create my male model, I sculpted the model in ZBrush and then retopologized it and made the UVs in Maya. Then I brought the model back into ZBrush where I baked the textures and used Decimation Master to bake the high poly to low poly. And I make the hair using XGen in Maya. I then painted the Adobe Substance Painter. I then used Adobe Substance Painter to paint on it and to show more of the physical symptoms of scleroderma. And then I made a fibroblast model in Maya. For the TRPC6 channel protein, I downloaded the model from the RCSB Protein Data Bank website. The PDB file was downloaded, and then I imported that to UCSF Chimera, where I cleaned the model and I saved it as an OBJ file. And then that OBJ file was then imported into Cinema 4D, and I used polygon reduction in order to reduce the number of polygons in the model. The reduced polygon model was then exported into Instant Meshes, which is a quad-based auto retopology program. And then I saved it as an OBJ where I then imported it for Maya for animation. And you can see here, I used Maya to create the 3D animations using blend shapes, and then exported the baked animations into Unreal. And the same thing with the TRPC6 model, which I rigged. I also wrote an audio script to help guide the pacing of the animation. And then I imported the animations into Unreal Engine. And there are a lot of issues that arose using Unreal and a lot of research that had to be done. But here is an example of how it looks like in Unreal and some of the research I had to do as well. And here are the blueprints I made. This is the blueprint that shows the starting main menu, as well as the box collision to start an animation. And finally, here is a result of how it looks like. Can you guys hear it?
The transient receptor potential cation 6 channel, also known as TRPC6, is a non-selective cation channel that is 6 times more permeable to calcium ions than sodium ions. The membrane protein is located in the lipid bilayer, where it acts as a signal transducer by altering the intracellular calcium concentration. The channel is made up of 4 subunits, A, B, C, and D, which together form a passageway for calcium ions via a pore between the 5th and 6th transmembrane domains. Let's see how we can alter the influx of calcium by affecting TRPC6 activity. By using specific activators and inhibitors that affect TRPC6 activity, the influx of calcium will be altered. Inhibitors that close TRPC6 channel include SAR7334 and laryxyl acetate, activators such as HYP9, PAMC20, and OAG open the channel, allowing an influx of calcium ions to pass through. Scleroderma is a systemic autoimmune disease that targets connective tissue producing cells called fibroblasts. This results in tightening of the skin, subcutaneous tissue loss, and telangiectasia due to activated fibroblasts that produce an excessive amount of collagen. However, research has shown that a specific channel may be involved in the development of this disease.
Right, hold on 1 second. There seems to be some technical difficulties. All right. That's it. That's the end of my presentation. Thank you.
Okay. Thank you, Joanne. That was awesome. Moving right along to our final presenter, and we're like right back on track time-wise, but I think we'll go ahead and still save any questions to the end, so jot them down or try to remember them. Our last presenter, Drisha Unitan.
Everybody, can you all see my screen? Okay,
Yep.
Cool. I'm a little nervous. Okay. So good evening, everyone. I am Drisha Unitan, and today I will be presenting my graduate thesis, which is cadherin-coded self-assembly of synthetic embryos, trial and error. My content advisor is Dr. Magdalena Zernica-Goetz, PhD, who works as a professor of Development and Stem Cells, University of Cambridge, and Bren Professor of Biology and Biological Engineering at California Institute of Technology. My thesis is based on the work done by Dr. Minbao, postdoctoral fellow at Dr. Magda's lab. Dr. Magda's lab is based on finding out the mysteries behind the embryological development and how a single cell leads to a whole human. Today, I'm going to cover the introduction, objectives, methods and materials used, the implementation, and the result that I've got, then the future work and acknowledgement. Let's first understand why is it important to understand embryology and why are synthetic embryos needed. The concept that your heart, lungs, brain, and entire body started out as one single cell is astounding. And whatever happens in an embryo's first few days of existence sets the foundation for a person's entire life. In the present day, due to increase in demand for fertility treatments and cryopreservation, embryology becomes an essential concept to provide insight into a mechanism of normal and abnormal development. It helps in comprehending the logic and process behind the organization of human body and aiding in the development of in vitro stem models. Dr. Magda has developed methods for tracking living embryos to determine how stem cells are first created, establish their fates, and work together to shape the body. The fertilization is when the sperm penetrates the ovum and the nuclei of these haploid cells fuse to form the diploid nucleus of the zygote. This initiates cleavage, a series of mitotic divisions to form blastomere. In the next stage, a fluid-filled cavity is created, blastocyst, and the cell mass is now called a blastocyst. Sorry, blastocele is the fluid-filled cavity and then forms the mass called as blastocyst. The early developmental stages for human embryo are self-organized and conserved. The cells are differentiated into 3 lineages from the start. The trophectoderm that will make the placenta from the outer cell mass, the primitive endoderm that will make the yolk sac, and the epiblast that will make all of the tissues of the future body form from the inner cell mass. The self-organization of these cell lineages is a crucial procedure assisted by cytoskeletons and the cadherins, which are the cell adhesion proteins. Recent studies by Dr. Minbao decode the cell type specific cadherin code that drives the differential adhesion lineage sorting. Here in the first image, we see human embryo throughout the early developmental stages of embryology. Cell sorting and cell adhesion is observed. The second image showcases the experiment carried out in which Dr. Minbao observed a set of stem cells with overexpressed cadherin and others which were wild-type stem cells. And in overexpressed cadherin stem cells, we see that they are well sorted. But the wild type, we observe that they are missorted. The result from this experiment was the cell sorting is triggered by differential adhesion among the cell types conferred by unique cadherin code. P cadherin in trophectoderm cells, E cadherin in epiblast cells, and K cadherin in primitive endoderm. The main objective was to create modules that are intuitive and engaging. The first objective was to create an interactive model to understand self-organization of the 3 primitive cell lineages of embryo. The second objective was to create 3D animation to enhance the understanding of how the cell adhesion is carried out in the presence of cadherin proteins. Now we look into the methodology and materials. I use textbooks, articles, and websites and webinars to research, webinars which were done by Dr. Magda to research on the topic. I took online courses on YouTube and LinkedIn Learning to understand Unity and Houdini, and even understand C++, that is the basis of Houdini. I took academic courses which helped me during the thesis, which were embryological visualization with James Perkins, particle dynamics concept in Interactive Media 1 by Craig Foster. The softwares that I used for my project are Adobe Photoshop and Adobe Illustrator for the 2D illustrations. 3D modeling was done mostly in Houdini and Blender and then were cleaned up in Maya and Mudbox. The protein models were taken up from PDB and cleaned up in Chimera. The interactive model was entirely built in Unity game engine, and After Effects were used for animation. So my implementation was carried out in 3 phases. The first phase was doing mock-ups, having rough diagrams and ideas recorded using Photoshop, Procreate, and Illustrator. During this phase, I determined the color scheme that I wanted to use. Since red, blue, and green were used for the cell lines throughout the research carried out by Dr. Minbao, I wanted to keep that intact so as not to confuse the audience. During my process, I came across many fluid simulations. I wanted to carry out cell divisions in Maya using particle dynamics, which was not happening, and hence I wanted to put in a slide that showcases the importance of trial and error in medical illustration, and in this case, trying out different softwares to figure out what would be the best for me. In the second phase, I started working on my models and animations. I figured Houdini was my best bet for doing particle dynamics, so I used the particle operations function. Junichiro Horikawa's YouTube videos helped me figure out the code that I wanted to use and the geometry nodes required to create these models. The first image is a screen grab showing the interface of Houdini and the primitive endoderm model. I used geometry nodes and personalized the C++ script to give me the cell division in that particular shape that endoderm cells divide. In the second one photo, the colored epiblast stem cell model using the same script, but incorporated a different shape. Here I use different sets of code since I didn't want the model to follow a particular pattern, and I let them freely divide. The first image is the trophectoderm, which is blue in color, and the second one is the embryo as a whole divided. Then second section of my phase 2, I started building the interactive models in Unity game engine. First we see is the rough sketch of how I wanted the interface to look like. The exported models, then is the exported models from Houdini, putting it in Maya to clean up and tried out different materials to see what would work. This is where one of the model is in Unity game engine, how it looks in the interface and how it was done. And below is the code provided by my good friend Pratik, who is a game developer. And the third part is the animations part, which I wanted to visualize the cell adhesion carried out by cadherins between the 2 cells, for which I used geometry nodes in Blender. In the third phase, I wanted to host the models and animations on a website so that it is an accessible learning experience for the audience. These are the frameworks I made to show how it would look once it's on the website. Now let's look into the result and conclusion section and see my progress on the thesis. After I built the nodes, this is how the endoderm cells looked like looked like on Houdini. And then after, you can see that. And this is how it looked after rendering. I need to refocus and learn a better way to render it out from Houdini so that the materials are visible and give a realistic experience. And this is how the trophectoderm cells were rendered. That's in the interface, and this is how it was rendered out from Houdini. And here is the trophectoderm cells dividing, and this is the rendered version of the trophectoderm cells. In the bottom left that you can see, this is the mis-sorting cells in Catherine knockout models that I wanted to show, which is a work in progress. It's still in the particle division phase. And this is the membrane structure that will be incorporated in the animation after I finish the better rendered versions of the cell division from Houdini. Now let's look at the interactive models that I have built. Okay, can you all see the screen? Okay, so this is, first is the day 2 epiblast in embryo. So I have added the zoom in, zoom out function, the x-axis, y-axis, z-axis rotation as well. Then on clicks, you will get the information on the model, and then even the protein that which one it's used for overexpression over here. This click for animation button I've given, once it's incorporated in the website, you will lead on to the animation once I have the rendered cell divisions ready. This is epiblast model. This one is trophectoderm. Same goes for that. I have all of these. Then we have the endoderm, zoom in, zoom out, develops into yolk sac, and this, and the protein that's overexpressed. Then we have a model that has all of the cells present, label them, and here all of the proteins were overexpressed, and is why the experiment was successful. And I have written a small information box that gives information about the experiment. Then in the knockout Catherine system where it was mis-sorted, and I have an information box here as well. All of these models have the zoom in, zoom out function and the interactive play. Okay, back to presentation. Oh, sorry. Yeah. So now the future work is to get better renders and editing of the animation for the cell divisions and integrating these animations with the interactive model, then using the WebGL platform to host it onto the website. And then I also want to do a standalone animation describing the project with voiceover. This would be done with the ending of the semester. And these are my references. And I would like to thank my faculty advisors, Jim, Craig, and Glenn, and my content advisor, Dr. Magdalena, and Dr. Minbao, and my friend Pratik for helping me out with Unity, and Junichiro Horikawa, who doesn't know my existence, but he helped me with my Houdini stuff, and my family, friends, and my supportive classmates. And I also want to thank the first-year grad students and the third-year undergrad students who are here. Thank you all for coming. Thank you.
Thank you very much, Trisha. Awesome job. Thank you, everybody, for joining us. We have some time for questions. In fact, we got right back on track time-wise. So anybody have any questions that they'd like to shout out? Feel free to just unmute yourself and just shout out or share them in the chat. And this always happens. People are always hesitant to be the first one. So I'm going to ask the first question just to break the ice. and try to kick things off here a little bit. Mary, I was curious. I thought your brain model looked beautiful. I thought that was great, and I'm really excited to see. I'd love to actually put on the headset and immerse myself. So I'll have to make sure I come to Imagine RIT and see your presentation. One thing I was curious about was how do you plan to focus on or extract the elements of the visual pathway? For example, something like the lateral geniculate nucleus. Would you simply make other parts of the brain transparent, highlight it, or can you actually potentially pull it out from the rest of the brain and isolate the visual pathways? What are your thoughts about that?
Right now, the plan is to ghost the structures within the brain and then toggle it to allow the users to pick what they want to see. So maybe incorporate a slider to adjust the opacity to make it transparent, and then manipulate it so that the user can zoom in and zoom out and see where everything is.
Just to reveal my ignorance about working in Unity, do you render like a completely separate model and then just kind of toggle back and forth between the visibility of the two, or are you simply like adding a highlight that you can sort of turn on and off?
So I am actually working in Unreal, not Unity.
Oh, sorry.
And what I plan on doing is I'll be creating the models in Maya and then importing it into Unreal. And then I'm going to have the help of a developer to code the back end development to connect all the nodes so that it's interactive and incorporate the slider and opacity.
Awesome. Cool. Thank you.
Yeah, thanks.
Just a quick follow-up question. Why Unreal and not Unity?
So I chose Unreal because I feel that it's more user-friendly for somebody that doesn't have a background in coding. I have, no background in coding, especially with the visual blueprint system. And when I did my initial research, it was the constant debate, like, should I use Unity or should I use Unreal? And I really do feel that Unreal is starting to take over Unity, especially being industry standard in like all the gaming engines like Matrix and The Mandalorian being completely rendered in Unreal. So that's another reason why. And I feel that it's a lot more realistic. And that was my main goal, to make a realistic brain.
I see a hand up. All right, Ari, is it?
Hi, I think Sheila might have raised her hand before me. Do you want to go first?
I had like a very student-y question. You guys did so amazing. And I mean, it's really, really inspiring to see what you all were able to do. I mean, it's incredible. So really, congrats. My question is, how, if you could like ballpark percentage, and this is for kind of everyone, what proportion of the work involved in your projects was sort of like stuff that software and things that you learned to use outside of the RIT curriculum?
I can start with answering that. So I ran into a couple of times, especially with my 2D animation, where there were things I didn't know. Like, I didn't know how to use Adobe Animate before I started this project. So I would say I focused more on the things that I already knew, like working in Maya for the 3D animation and developing backgrounds and that kind of thing. And then anytime I had left, I would spend focusing on new stuff and trying to add more to what I had.
Yeah, I can add on top of that. I think RIT provides a lot of resources as a technology school, so it's up to you to go and find it. A lot of these things that I've learned were from other courses besides the one in the medical illustration program. For example, I took a 3D modeling character design class with David Sanchez, who also was kind enough to be one of my thesis advisors. But with him, I learned how to really use ZBrush and Adobe Substance Painter and all those other programs you saw me use.
Anybody else want to jump in before we move on? Yeah, go ahead, Tracy.
Been taught in the curriculum, but what I've observed is since we learn Maya, it sets like a background, solid background for us to learn other 3D softwares. I found it easier to go through, surf through them since we have a strong Maya background. So, and all of these softwares that I've used, all of it is found on the computers that's there in the medical illustration lab studio. And Craig is always there to help you with the 3D stuff. And he's always been like around fixing stuff that I, have gone wrong. So yeah, I feel like you can still use the software that we are learning, but other softwares are easier to learn as well.
Yeah, to answer your question, Sheila, I agree with Trisha and Joanne. I feel like for my project, especially, I learned a lot of new software, like ZBrush is my first time using ZBrush, my first time using Unreal, my first time using Substance Painter. And I took the same route as Joanne to take 3D modeling mastery with David Sanchez. And he also knows a lot of Unreal Engine too. So we were able to kill two birds with one stone with that. So the resources are there. It's just up to you to pick and choose. And yeah, I would advise you to, figure out what you want to learn. And I think something I would do differently is, get started a lot earlier, like during the summer, and then make those connections. RIT is there, and just knock on their door and be like, hey, like I want to learn this. Are you offering any classes? And that would be really helpful.
Excellent advice, Mary. Start early.
I have advice on the flip side, because I chose a thesis based upon not necessarily learning a bunch of new things. I just kind of stuck with the basis. That's also something that, is a perfectly viable option. I mean, yes, I do see a lot of the things that I've created personally in my thesis as assets that then could be used in animatics or things like that, so they could be expanded upon. So it's just kind of one of those things of like, don't feel like you need to like go out and be crazy and kill yourself with a bunch of different programs and stuff. I mean, it's great to learn it. Definitely don't discourage it, but you can also keep it within that means too. So that conflicts with everyone else. I feel like it's valid too, Yeah,
thank you.
All right, I think everybody had a chance to chime in. Hopefully I didn't miss any other hands. I didn't see that one pop up. So Ari, your turn now.
Hi, congrats everybody. You've all done such amazing work. I'm so impressed. My question is for the two folks who did virtual reality projects. One of the sort of risks associated with learning in virtual reality is the possibility for motion sickness. And I'm wondering if there are any design elements or artistic sort of strategies that can reduce or mitigate that risk?
Joanne, do you want to go first? Do you have an answer for that?
Yeah, so there is research out there to help reduce motion sickness. I haven't really paid really attention to that kind of design aspect, but the way that I feel like I'm approaching it is incorporating like animations, so that way that people have like time to like readjust. And I know in certain games, that in VR games, you can teleport into your location instead of moving around, which reduces motion. But for right now, I haven't really incorporated that aspect into my project.
So I can totally relate with the motion sickness, because I've played a bunch of VR games and I just wanted to throw up and I just couldn't take it anymore. So when designing my app, the way I designed it to reduce that is that the user is going to be in a fixed place. So there is no teleportation or any type of sliding movement around the lab. It's just going to be the user standing there or sitting there and then just, interacting with the lab. So that's how I plan on getting rid of that aspect and preventing that.
Just to chime in on that real quick, there was a presentation years ago at one of the Association of Medical Illustrators meetings. Craig, you might remember this. I think it was some of the isoform guys. And one thing that always stuck with me from their presentation was that a lot of motion sickness comes from movement of objects in the periphery, in your peripheral vision. And so blurring out objects in your peripheral vision can help to mitigate some of that motion sickness. And I see David nodding his head down there. So
Thank you.
any other questions? We have a lot of congratulatory comments in the chat. I'm not seeing any additional questions there. So other questions that folks want to shout out or raise a hand? Hopefully I won't miss you.
Just one quick question for the VR guys. Have you found any research that suggests there's some kind of loss of understanding because there's not a tactile experience involved? You know, I've seen a couple of virtual labs now for dissections and for the kind of anatomy that you're studying, but I was just curious, has anyone actually looked at loss of actual physical contact with an object versus just being able to look at it virtually? Is there any difference in retention from the learner? Or I don't even know if that's out there. I'm just curious if you came across it.
Well, from my literature search, what I found is that a lot of the virtual reality tools, the educational tools, were effective as being supplementary material and not to completely replace it.
Okay.
And I also found that haptic feedback is really important. So if you were to, so for example, for a dissection, if you were to touch like the cadaver, then it would vibrate, right? So that would indicate physical touch. So there's different ways of imitating that, but the main thing is that it's not supposed to completely replace learning.
Thanks.
Yeah, I agree with Mary. Having like haptic feedback, having a lot of immediate feedback is really important to improve learning, as well as making sure that there's not a cognitive overload. Basically, because you are in this like very realistic setting, you are taking in a lot more visual information and stimuli. So making sure you don't like overburden the users is really important for them to maintain the working memory.
And to add on to that, it's really interesting because a lot of different researchers at RIT that are researching haptic feedback, it's not just limited to just vibration. There are some devices attached to the headset that might imitate like water being splashed to your face, and even temperature, like cold and hot. So I think that in the future, that's definitely going to be implemented with education in VR to mimic that.
Thanks.
Who else? Anybody?
I have one more question for Drisha. Specifically, I think it's really interesting that you were able to make the 3D models of some like genetic manipulations of coherence, if I'm remembering correctly. Do you see your modeling being able to like sort of expand, and like what you showed was like kind of the science informing the art, but since you're also officially a scientist, can you see your art and your molecular modeling driving forward any new interesting research directions?
Talking me? What is it about my stuff? Okay, cool. I don't actually, because I am not working into the science section of it, just focusing on the art section of modeling and just like showcasing what the experiment was based on. No, I've left my scientist background.
I want you to continue. I think it's really, really interesting. And I love.
It's really interesting, yeah. But I do want to focus on being a medical illustrator.
Yeah. But I love the idea that, you know, from the research side, that there could be something tangible instead of just, the one image that you were able to get from like a confocal microscope image. Like, I think this could be like a highly, sought after type thing for researchers.
You
not
should
just
you should
embryology.
look into my content advisor's work, because she has now reached where she has built a functioning heart from synthetic stem cell. So yeah, you should check out her work, and you would really love that, because it's pretty good. Yeah.
I think you should also make some for like make a cell and every molecule you can see. I really like it. So yeah, thank you. I will check it out.
Thank you.
You definitely don't have to leave the scientist side behind. I mean, there are plenty of people who do both equally well. I'm thinking, I mean, I keep talking about David Goodsell in the scientific visualization class. Now he's a bit unique, but I mean, there's people like Gail McGill at Harvard. He runs a couple of companies, Digizyme, Clarify, that do this kind of very sophisticated animation work, but grounded in real hard science. He's trained a number of students who've gone on. Janet Owasa, I think she's out at University of Utah. People who are doing, a lot of very sophisticated science and this kind of visualization work at the same time. So now,
Okay.
if you want to just focus on the art side, that's fine. That's personal choice, but don't feel like you have to give up that side of it.
All righty.
Yep. I think you're muted.
Good to see you, Jim. Good to see all of you all. The graduate student projects are really impressive. I'm pretty old school. I go back to 1981. So the concept development is the part that I really enjoyed hearing about, because these are all such complex projects. But when you get involved in all the different new media and the 3D development, and especially in augmented reality, I'd like to know from the students how they kept from becoming overwhelmed while they were teaching themselves all these different programs at one time. Can anyone comment on that?
Excitement.
Oh.
Yeah, it's interesting, right? Like it's so new and it's intriguing, and there's just so much to learn. So I think that's what really motivated me. It just kept opening doors, like, oh, I didn't know this existed. So yeah, excitement.
Oh, good. Did you use any online learning yourself that you found very effective?
Yes, lots of YouTube videos, lots of LinkedIn learning, blogs, Reddit.
Well, one thing I'd like to say is that after working a long time at the College of Vet Med at University of Georgia, one unrealized objective that we had was the use of augmented reality for patient education. There are a lot of hospitals that you can imagine would have, a lot of empty hallways where you could actually put up large posters and on things like diabetes and other subjects that would be relevant to the patients within a particular area of that hospital. And you could turn that into a target and then use these skills that you've developed to create, an animation or an interactive program that actually goes with that poster. So I feel like what you guys are learning is tremendous. I wish I'd had this kind of experience when I was in school, but this technology didn't exist. It's really impressive to see what you guys are doing with it. And there's a great application for it. So I think that when you get around to applying for your first job, that all this experience is really going to come in handy for you. And so what I would like to do is I would love to see these projects when they're finished. Will there be a place that I can look for these online if there's a way to access them? I'd love to be able to see them. I guess, Jim, that's for you. Is that a question?
I'm looking at Craig on my screen over here. That sounds like a technology thing for Craig or Glenn. So I'm going to pass the buck, but I think that's a great idea.
Yeah, and we'll keep you posted, Kip. I know a lot of the students are planning an online component at least, so that'd be a great way for you to be able to access their work. So once we start seeing the final thesis projects come out, we'll let you know.
We can always open the Kip Carter School for Interactive Learning.
Yeah, right. No, but when they're done, I do go to the university occasionally, and they're still having their Friday 3D user group, and these would be great topics to take a look when they're done. So we'd love to do that.
And to follow up on something that Kip mentioned, the idea of having like posters in the hallway with QR codes or whatever, and pulling up some augmented reality stuff. We have Gary Welch, one of our alumni, has joined us. He's hidden at the moment, but he came and gave a presentation on campus here a few years ago, and I remember he had something exactly like that. He had some big posters that he unveiled and held it. You can hold up was Gary, was it a VR thing you'd hold up? Was it the headset, or was it just a phone, or I can't remember now.
Yeah, there was like iPad, and there'd be a poster with the QR code, and then it would trigger the experience, and it'd be like you're looking into a hole in the wall. You could like play a game in that hole in the wall or whatever, or learn about anatomy or whatever,
But another great application for that we found was in scientific presentations, because you could do the same thing with a scientific poster. And our clients really love to be able to stand out in the crowd in that way. And we were using either videos of them talking about their research, or we do a 3D animation that explained what their process was about. So there's a lot of applications for augmented reality out there, I think.
That's a great idea, Kim. Just because I love molecular stuff, I could just imagine you have a scientific poster and you've got, like Joanne's, what was it, TRPC6 or something, and hold up a QR code and you bring up a 3D model of that exact protein that you can manipulate around and explore it in 3D. Yeah. So, cool idea. Heather, you have your hand up.
Hello. Want to hop in and say, I graduated from RIT in medical illustration. I'm still doing medical illustration. I have my CMI and I'm starting medical school. So please don't give up on doing both medicine and art if that's something that you want to do, first off. But I do have another question. I know that this is something that I've spoken with Danielle about, with the rising cost of medical illustration programs, was looking for something that is open source and free to use on the list of priority for you guys when you are looking for new programs to use for your projects, because we've discussed that a lot of you went and used new programs with the trending increase in cost and sometimes decrease in pay for some of these projects. What thought process did you guys go through when you were picking those programs, and was that a part of it?
I can go for this question. So most of the softwares that I've used are open source. But yeah, so that went into my thought process where I was just like, oh yeah, I need to use something that's free. I don't want to spend money. So it's not like I'm spending $5. It's very expensive, all of these softwares. So, but for the softwares that I needed to pay, I used to just come to campus and work, because campus has all of these softwares, all of the educational license for these softwares. So I would just come in and work on them, or else I would choose, free softwares.
And even in teaching, I mean, it's one reason why in the scientific visualization class, I've always tried to find free things, not to save RIT money so much as to allow the students the opportunity to download these things and use them for free also. So, Chimera and PyMole, Osirix, Horos, 3D Slicer, these are all open source tools that we use in class, and then students can access on their own. We are at just after 7:30, so thank you everybody for staying on so long. Do we have any other questions before we start to celebrate?